look, thanks very much for attending this evening's presentation. We're really excited to have Dr. Richard Lobb present to you and to us an update and a progress on the research that the charity has funded from our most recent event last year, which was the 10-year anniversary of the Bondi to Berry bike ride. My name is Nick Young. I'm the co-founder and the chair of the Dementia Research Community. Now, without any further ado, I will introduce Dr. Richard Lobb, University of Queensland, with our most recent research grant that was provided last year, and I'll welcome you now to present.
Right, perfect. So, yeah, thanks again for the introduction. It's great to be here. Really happy to be able to share the work that we're doing here at University of Queensland and what my project is going to be looking at and how we're going to advance the, sort of blood testing in the diagnostic space for a variety of diseases, including CTE-related dementia. So the title of my talk today is a bit sciencey, but it is the title of my project, where it's a simple blood test to spot chronic traumatic encephalopathy related dementia early. So the idea about that is being able to take a blood test and looking at their blood, what sort of signals are in their blood, and being able to diagnose the test, getting that molecular information on diseases instead of just relying on imaging. So this work is really only possible because of this funding grant that I've been awarded. So thank you very much for the funding for this, because this is going to be able to advance the space. So just a little bit of background. So in this presentation today, I'm going to give a background of who I am, what my research is, the scope and the vision of what we're going to be doing, to give you an idea of where we're going to be going over the next year. So I'm a NHMRC Emerging Leadership Fellow at AIBN. So this is the AIBN here, based at the University of Queensland. So the main work that we do here is sort of nanotechnology, where we just design devices that are really capable, highly sensitive readouts for advancing the diagnostic space in a variety of diseases. So I lead the EV Diagnostics and Therapeutic theme at the Centre for Personalised Nanomedicine based at the AIBN. A bit of introduction of what I'm going to be working on. So this is the CTE, which is really a progressive disorder that's a result from traumatic head injury. So this could be in a lot of sports, military service, or accidents can result in this. At many levels of society have experienced this. One of the main important parts of this, though, is that you can only confirm this after death. There's no way to actually determine if someone is suffering from CTE like fully 100% until a post-mortem biopsy is done of the brain. So the feature of this is that you get this accumulation of this certain protein in these cells, and where it starts is a little bit different than Alzheimer's, which a lot of you will be familiar with. So it's very similar, but it has some distinct pathologies as well. And so what we're trying to understand is that, can we look at this specific form of dementia related to CTE, and then eventually we'd be expanding this into all sorts of diseases, including Alzheimer's for that. So what does the CTE looks like? Well, it's divided into clinically into 4 stages, where you have these early stages where you get that certain protein that I mentioned, and it goes through these advanced stages where the whole brain is affected. So what that looks like when you look under the microscope into these cells, these brown spots here, you can see these are the affected cells that are accumulating that toxic protein in the cells. Stage 1, you can see that there's not so much of this brown staining in these cells. You get more of that brown staining, and then as you progress in later stages, you can see that most of those cells are expressing this toxic protein for that. So as I mentioned in the earlier slide, the only way of actually diagnosing this is post-mortem. So what we're trying to do here is develop that new approach for that. So you can look at conventional approaches for all sorts of dementia-related problems. Maybe you get imaging, or eventually you might get tissue biopsies. This is really kind of a reactive approach. It's not equitable to a lot of Australians anyway. It's slow, and it's a delayed readout. Needs a lot of dedicated infrastructure. So the approach that I'm proposing here for this project that's being funded is that we're going to be taking a blood test, where you just take a simple blood test. You have a routine GP visit eventually. And that's going to reduce that infrastructure requirement for those advanced imaging facilities. It's also a thing that's very suitable for rural and regional Australians as well, because they don't need to go into metropolitan areas. They could just get a blood test and get all this information. And so what we're doing here is developing that new technology where we're capturing what's called extracellular vesicles, or EVs, and I'll tell you a bit more about that later, which is what this project's based on. So provide what we call a window to the brain. So instead of relying on sort of indirect measurements or late stage readouts with imaging or biopsies, we're going to get real-time information and early detection. It's really proactive in its way that's going to be doing. It's equitable for all Australians, and it's inexpensive and fast. So there's a lot of advantages to using this approach here. So what you can see there as well is that these are the types of nanotechnology devices that we're using. And so I think I'm just going to, this is kind of what that looks like in real life. So you have a picture there. We just have this gold-plated nanotechnology device here, which captures these signals specifically from the blood, where we can read out with really high sensitivity based approaches there. So just a sort of case study of how this does work for those that are interested. So we've recently just published this approach in brain cancer. So the Mark Hughes Foundation has funded our work in glioblastoma, which is a highly deadly disease there. And we can use this device to capture those signals coming specifically from the brain of these patients, and before imaging, determine if they're progressing in the disease at all as well. So it provides a really nice new window into the brain where we can get signals that weren't before. So now the plan is to take this into the dementia space there. Can we get this approach really working specifically and reading out dementia-related signals from the blood for that? So I've been mentioning EVs, and some of you might be sort of sitting there going, why is this guy talking about electric vehicles? We don't need to know about the next Elon Musk related story, but EVs are not electric vehicles in this sense, but actually what I mentioned is extracellular vesicles. So I know this is a bit sciencey, but you could consider these are extracellular, so they're outside of the cells, and they act like text messages in between cells. So they're these little packages that contain a lot of information, and cells use them to send messages from one cell to the other. Importantly, they'll actually come out of the brain and get into your blood where we can detect them. So these messages are 40 to 150 nanometers in diameter, and they might not mean much, but if you talk about a grain of sand, that's 62,500 nanometers, right? So there's really, really tiny messages, but as a text message, they can contain a lot of information, and they can reflect, importantly, it reflects the disease that these cells are from. So when we're looking at these little text messages that are coming out of the brain, they get into our blood system, and they reflect the cells that they're from. So you could have your neurons in your brain that you might be familiar with, or your other supporting cells in the brain. They reflect that cell that it's from. So it's like a little copy going around in your circulation, which we can then pull out specifically and interrogate. And so these little signals there, that sort of window into the brain, could be from those different cells that can show signatures of that disease that you're specifically looking at. And importantly, once you get eyes on this, right, you can start looking at therapeutic approaches, because you can get real-time readout. Is a therapy working? Instead of a late stage, you just find out before you can change any therapeutic approach there. So it's really going to help in new therapies coming on. Can we get a readout? Is this helping earlier than waiting for too long for that? And so this kind of really feeds into what we really know about the needle in the haystack. We've all heard that sort of problem here. So we have these EV markers that are in the bloodstream, and we're trying to find that needle in the haystack. When we're looking at EVs from the brain, that becomes even a high resolution. So we want to take out that specific population and sort of make it a bit brighter, if you want. But then we're looking for EVs from those specific brain cells, so from the damaged neurons or the specific supporting cells. It is really like we need to get that precise needle, find that exact needle that we're after. And the way that we're doing that is that in that device that I showed you. So what we can do from that is actually start making that needle stick out really well, make it very easy to find. So the idea from using this device to capture those specific EV signals from the different brain cells, really highlighting what it is in that haystack and finding it. So we actually have all these billions of these different EVs in our circulation, sending messages from one cell to another. What's the right population? And in the way that we're doing it there is how we kind of term it. It's a multi-factor authentication approach. So we're all very familiar with multi-factor authentication in our daily lives now. So it's how we identified as the exact individual we are amongst the several billion people on this planet. So you might be looking at multi-factor authentication, we globally, the country-specific signal, the city that we're in, the IP phone passwords, and that can identify us as the individual who we are. When you're talking about these EVs from the brain and the circulation, it's the same kind of approach, and our devices allow us to do that. So you have all those blood-based markers that are there. We can then zoom into specific tissue or organs like the brain. Then we can then zoom into specific cell types, so those damaged neurons, and then eventually get that very specific disease-specific EV readout. So we're only looking at that, getting what you would call a GPS signal directly from the brain, and being able to monitor the pathology that way. So in this approach that we're doing for this project here, we're going to be looking at these neurons and astrocytes and microglia. That's just the cells of the brain that are damaged in this CT-related pathology. And using that sort of multi-factor authentication where we're capturing that specific EV population, reading out that disease phenotype of what cells it's from, and we can verify that as a very precise GPS signal. And this is with my team up here at UQ, also guided by Professor Matt Trow, and my PhD student, Shuming Yu here as well. So overall, the sort of vision and expected outcomes of this project that we're in the very early stages of getting going is that we're going to develop a simple blood test that can really flag these changes very early before these problems become obvious. And how we're doing that is that I showed you that device that we're going to be using to capture those signals to get a really clear picture of what's going on in that individual. I guess the biggest scope for this as well is we're going to move this into other dementias too. So Alzheimer's, we can really start expanding the scope once we've started getting this project up and running in the CTE space. And then the sort of larger vision here is that to take this into sort of MRFF, like really start leveraging the funding to scale this up into from discovery into real world use and actually get it in the clinic where it can become just a regular GP test that you go to for the doctor and monitor these things and get early detection instead of late stage detection. So just want to acknowledgements as well. So my team up here at UQ, also Fatima, who I'm collaborating with on this project, and my funding, of course, Dementia Research Community here. Really appreciate the support that you're going to be giving this project. I think it's a really great chance to drive highly sensitive diagnostics in this space, and my NHMRC fellowship. So thank you very much.
Wonderful, Richard. Thanks for that high-level summary and explaining that in some terminology that for me was a bit easier to understand. I know you've got pages and pages and pages of theory behind this one, but that was a really nice, easy to follow presentation. I just wanted to ask anyone on the call, are there any questions specifically that anyone had raised during this presentation or the research that Richard's just giving us some insight into?
It's Catherine here. I'd like, it's a really good presentation, Richard. Thank you so much. I'm just interested, would this technology, the nanotechnology with the extracellular signals going, how do you see that working in with the existing blood-based biomarkers with p-tau217? Seems to be able to, that's coming up, and 181. Is it something that you think this technology is more specific than those approaches?
Yeah, that's a great question, right? There's a couple of points that I think there are advantages from this, right? So It's these extracellular signals that are actively released by cells. So it's a really active release process where they're actively released. And so we have you might be familiar with the blood-brain barrier. So that really specific cell layer there, which prevents a lot of signals from coming out of the brain, unless that's sort of broken down a little bit, which does happen, but not really early on, right? Or there's not a lot of evidence that happens very early on. So these EVs, they're actually actively transported across the blood-brain barrier. So they use their own biology to be transported across, where you can get them in the in your peripheral blood or your circulation from that. So some of these other biomarkers, they might not really appear at early stage. And I think that's the key difference here, is that you can get this signal at the earliest stages. So we've done this a lot in cancers, where we can detect this at the earliest stage of cancer, when the tumor is at stage 1. So, and also in the brain space, when the brain cancer is only just coming back, and so there's only a few cells secreting us into the circulation. Likewise, the hypothesis here would be in the dementia space, you're getting that signal very, very early on, and it's specific. You can also look at a more broader range, so how your body is reacting to those damaged cells. So we have our own immune cells in the brain. We can take those signals as well and look at how we're responding for that.
No, good, very good.
Thanks, Catherine, for that question. Does anyone else have anything to ask while we've got this opportunity?
Yes, please, I do. Thanks again, Richard. That was really interesting. Something I was wondering is, how advanced do the symptoms of dementia need to be before the EV can actually identify there's a problem? Would it be before there's any symptomatic problems going on at all, or would it be later on or more advanced dementia?
Another spot on, great question. It's the answer to that is that that's not really known, right? And this project is going to allow us to develop how early you can determine that in humans. So it's really, I would say, it's more of this sort of cutting edge science that's being done right at the forefront to really start elucidating those answers. And so the idea behind the project is that you do want to be able to capture these signals before you have these advanced clinical manifestations of the disease. So can you start detecting this before that stage? And so my answer is that I would expect so. That is what we're after for that, but the evidence is not in, and this project is really going to start helping provide that evidence, if you want.
But they would have the potential to do asymptomatic general screening of the population prior to any dementia actually developing?
Yes, I think ideally you're going to be wanting to go into space for, if it's going to be used for what I mentioned, a screening or a blood test, that you do have that asymptomatic stage, but it could obviously be used in both. And but it's really the idea about using these really precise sensitive technologies combined with those extracellular vesicles, which give you that cell specific readout from where that disease is coming from, that you then get that early detection that hasn't really been possible yet.
Thank you.
Sorry, I have another question for you, Richard. I'm just thinking about, obviously, this particular blood spot you're looking at, CTE. So we get patients that come in that have a history of head injury, perhaps not consistently. So presenting as potentially Alzheimer's. They've been diagnosed, they're on perhaps, current standard treatments, and then having the blood-based biomarkers for Alzheimer's. And do you think that there could be perhaps that opportunity where you can do, the dual approaches to see exactly what their pathology underlying is? And secondly, there's another question there with it too, is I'm thinking, is this an opportunity where you would like to have, a clean baseline from perhaps younger people who haven't yet had any head injuries to be able to go, this is comparative, this is the way they are now? Is this something that you could track over a period of time to see any impact?
Yeah, I think in terms of some of the clinical samples that we would be wanting to run, sorry, answer your second question first. Yes, so there would be healthy controls there, which are age matched, and there are some in terms of these CTE suspected cases that at an early stage from these contact sports. So yes, age matching for that, longitudinally tracking these people as well. So that's going to be a really key part in expanding the project, is that longitudinal monitoring. And this is also what this the whole idea about this grant as well, is to start longitudinally tracking people in terms of that. So do we see that change over time? And as in terms of the biobank samples that we have access to, we'll be able to get that close to before death, where they really start developing quite significant features of the disease for that. So in terms of the first question, were you asking in terms of the blood-based testing, would we be able to distinguish between Alzheimer's disease and CTE? Is that?
Yeah, I mean, a lot of the times it's sort of taking on board, I guess, the clinical presentation and the degree of trauma perhaps that someone's experienced. But I'm just thinking that it's likely that there could be this could be the trigger for the pathology that then becomes sort of multi approach to sort of disease, multimodal. And there's not very many cases like that where you sort of wonder, but obviously as you develop this is an opportunity to use multiple different approaches for defining the diagnosis.
Yeah, no, I think you're spot on there in terms of that. Being able to differentiate the two at that stage might be a little bit difficult, but we're going to see whether there is key differences in that. But yes, I think where you could go is to help sort of start in terms of the sort of holistic approach of all the information that's coming in with the other approaches, combine that as well. And I know it's a buzzword, and I'm kind of getting a little bit tired of it myself, but when you do integrate this with AI measures as well, you can start being able to really improve that kind of classification, I think.
Great.
Yeah, good questions there, Catherine. I know at Caramind you're a really strong advocate for baseline testing, and if this is a blood test that can be rolled out at a certain age, obviously before impact to the head or the brain, that's a really good fallback and a baseline to work towards in the future should things start to deteriorate. Thanks for the discussion so far. Does anyone else want to ask any questions of Richard or the research?
It's Tom here. I might just jump in and just ask more broadly, with the success of this project, with how you're hoping that it should play out, do you see this being commercially very successful and viable, whether it's venture capital or whatever it might be, to be able to apply this to professional sports more broadly? Being able to take that baseline and say, as we were talking to before, you've got your rookies coming into a professional sporting league. This is part of their sort of medical panel or medical that they get done before being a contracted professional athlete, and something that's measured over time.
From a scientific perspective, I think absolutely, Tom. I think it would be essential to look at people's health in sport, right? I mean, I love watching the footy myself. I'm a big fan of that, but there is no secret that, if you look at the history of NFL, right, and ice hockey in America and where that's come from, and we also having rugby league players now that have got probable CTE, and there's a lot of problems going on there. It's going to become more of a really important topic in terms of player management. So I think from that perspective, it should absolutely be done. It would be more, I guess, sort of engaging with those stakeholders, whether those, and I know there is some appetite for it, but how far do they want to take it in terms of monitoring their own players? Because if you're getting all this information, which is saying, no, you can't put them on the field, there might be a little bit of kickback from that, right? So I think in terms of a health perspective, that should be absolutely done. And I do think from a standpoint where they're managing the players, that those, from my sort of initial conversations, that there is definitely interest in that. And so to drive that, it does need that commercial industry aspect for it, to drive these translational applications, and particularly in the contact sports space. But so kind of two-part there is that, yes, it should definitely be done. It would be interesting to see how far those organizations, how much information they would want to get, and I guess further engagement with them to find that out would be sort of appropriate for that. But that answers your question.
Yeah, absolutely. I just think that I've had a few family members play through the AFL, being Melbourne-based, but where they've had significant head traumas, which is now showing in their 30s, caused early retirements and so forth. And I think that organisations like the AFL Players Association, the AFLPA, they're very much on the forefront of player wellness and health and well-being. And so they're the types of organisations that I think from a commercial aspect, if they can have something like this in their arsenal to be able to track how players are and their well-being, would be absolutely of highest importance, which I think, any player associations around the world would see the same.
Yeah, I think as well, just to follow up there, Tom, is that and in terms of expanding the scope of this project as well, this does give us the chance to really get that preliminary data where we can go to those organizations and then partner with them in terms of the MRFF funding, which you can then leverage. So there's schemes there that you can have partners come if they've got co-funding for that, then the NHMRC MRFF will match that, and you can sort of grow that into a very large project and start sort of developing that to eventually develop that commercial platform that can be used in that sporting sense.
Good. Thanks, Richard. Thank you, everyone. I think unless there's anyone who wants to put their hand up very quickly for a question, we might start to just summarise and wrap up. Just to acknowledge again, Dr. Richard Lobb from the University of Queensland. So receiving funding from our grant program last year, and we're very much looking forward to hearing the progress of this one as you move forward with some of this testing. And you were the 12th recipient of grant funding from our organisation, and I think we are upwards and moving forward on this one. And just to acknowledge again some of our sponsors and partners that have joined this call. I think this goes a long way from our perspective to show you that your support financially and in kind goes directly to the research, and we've obviously had the benefit of being able to hear from Richard Lobb tonight. In due course, do some introductions along the way to our valued partners who are on the line here, and also some of our supporters. Unless there's anything else, I might wrap up with a final plug and a shameless promotion of the 14th of March, which is our gala dinner here in Sydney. It's the 10-year anniversary of our charity, and we are hosting a gala dinner which is nearly sold out. So if anyone has anyone who wants to attend, I know a few of you on the screen here are attending, and we thank you for that. We will be announcing our 13th research project on the night, which is excellent. And depending on how things go with auctions and raffle tickets, hopefully a 14th research project, or at least put our hand up to say, with the generosity of everyone on the night, we will be moving towards a 14th research project. So we are really excited about the future of our charity and our organisation. Now that we've come 10 years, we've got some really good basis and foundation with our board and committee, and looking forward to what the next 10 years bring.
I just want to say as well, thank you for having me, and thank you for the funding. It's given us the opportunity to really delve into this a lot more, where we can start expanding this space. And I'm very excited about the project ahead and what we can do here.
Yeah, thank you again. Thanks, everyone. And we'll keep you updated on this progress through our supporter newsletters.